Hey guys, it's Mr. Haas here, and I want to walk you through this online virtual microscope that we have here. And I know it's not the same as being in class, but with you being virtual, this is the closest we can get to the microscopes that we have in class. So this is a very lifelike microscope activity. So you're kind of on your own here, but I do want to share a few things with you. There are some buttons here that can help you. If you press guide, it'll talk to you about the different terminology and how to take care of a microscope. So take a minute to kind of take a look through that. So I'd like you to do that first, okay? I know it's not the most fun part of it, and I know you want to just jump right in, but take a look at that first. Then go to the learn part. And for the learn part, it says select parts of the microscope to learn more about each part. So you're going to have to use these. So this is the actual virtual microscope you're going to be using. So it makes sense to take a minute to click on the different buttons and the different things. So if I click, hey, what's this? Oh, it's an eyepiece lens, and it tells you a little bit about it. Okay, then you can click that, and it's the coarse focus knob. And it'll tell you a little bit about that. So make sure you take a look at each one of those and click back to main when you are ready. The next thing I'd like you to do is then the fun part. That's what you're looking for, and that's the explore part. So when you click on the explore part, it's going to bring you to the microscope. So the first thing that you have to do is scroll over and click on the little arrow, and then it gives you the slides. So we have some sample slides, plant slides, animal slides, bacteria slides, and human slides. So we can, I'm just going to show you one. So we're going to pick a plant slide, and underneath that, let's do a plant cell. So it's going to pull it out for you. And so what you're seeing over here is the view as if you were looking in there. So here are your different focuses, your different, magnifications. So we always start with the 4x. That is your scanning objective. That's the lowest one, which allows you to see the most so you can find the specimen. So it doesn't look like a lot at first. So you want to go down here where it says coarse focus, and if you take a look as you start to do the coarse focus, if you go back and forth real quick, you'll see that the stage rises up and down just like it would in a real microscope. So the coarse focus is your, that's where you want to get the image pretty clear. So I can see it, I'd say probably right about there. And then I grab the fine focus and pull it until it is like crystal clear, which I think is right about there. You can adjust the light if you want. It does change it a little bit, the contrast, so find what looks best to you. And then there you go. So that's an example of a plant cell. So pay close attention to what you can see, because you're going to want to compare it to the other organisms that you see under a microscope. Then we have you can go to the next objective, and that's your 10 times one, and you want to go in order. So now we're looking at 10 times bigger here, and you can see that they got bigger, but we can definitely tell this is a plant cell because the cells are rectangular. And remember, each one of these little brick looking things are a cell. So this is a plant cell is definitely multicellular because there's multiple cells all stacked together in there. We can go to the 40 times one, and you might have to adjust a little bit the focuses, so play around with it. Not the coarse one so much, but the fine one you might. And now here you can actually see the cell looks way bigger, and you can see the little nucleus and the chlorophyll. These little green modules in here are the chlorophyll, which is what gives the plant that greenish color and allows it to take sunlight and turn it into usable energy. And the nucleus is like the brain of the cell. And then lastly, for the 100, when you try to go to 100, it's going to say add immersion oil. So just go over to the little question mark, click it. It's going to put some oil on it so that you don't scrape the lens. And you might want to adjust the light. And then we can really see that nucleus of that cell. You got to be careful not to do the coarse focus too much, or it's going to tell you that you damaged the microscope, and we don't want that to happen. So just like real life. So just go ahead and hit done, and it'll fix it for you. And then you got to go ahead and carefully find it again. And there you go. So I want you to explore it. Just have some fun with it. And when you're done with this slide, hit remove slide. You got to use the lens paper to clean. The immersion oil off. It takes it back. And now you can go into the slides and do another one. So when you're all done, you can get out of here, and it should allow you to go back to the main. And then you can click the test if you'd like to see how well you can answer some of the questions. So have a lot of fun, enjoy it, and good luck.